Hello, so welcome back to your own channel of Curious Corners in Biology, where we discuss and dive deep into this subject. We discuss the scientific inquiry into this subject, make complex topics very simple to understand, and induce curiosity in the minds of learners. So, this is the objective of this channel. As usual, today again we are going to discuss something interesting. We are going to discuss something about Golgi ribbon. Okay. So what is Golgi ribbon and why is it organized? How is it unorganized? And why it needs to be unorganized? Okay. So in common text, we are familiar with a cell organelle that is Golgi complex. This is also called as Golgi body. This is also called as idiosome. This is also called as Baker's body, Golgi apparatus. We are familiar at least with Golgi body and Golgi apparatus. But then what is Golgi ribbon? Golgi ribbon is an interesting structure which is usually found in vertebrate cells. In vertebrate cells, usually you will find Golgi ribbon as many Golgi complexes connected with each other through tubules. Now, let us first review what is Golgi complex. Golgi complex is a cell organelle which is found in eukaryotic cells, and it has cisternae, it has tubules, it has vesicles, it has Golgi and vacuoles. There is a whole anatomy of this organelle. But the main function of this organelle is to receive substances from endoplasmic reticulum along its cis phase, which is also called as loading phase, apply all the chemical modifications and release the modified chemical substances through the trans phase into intracellular targets or extracellular targets. Even the proteins from ribosomes are also modified in Golgi complex. So this is a broad summary of the function of Golgi complex. But then if different Golgi complexes associate with each other through tubules, then we get what is called as Golgi ribbon. And let me share my screen and show you what is Golgi ribbon. So on this screen, you will be able to see Golgi ribbon. Okay. So here on the top, Golgi apparatus has been shown. Cisternae has been shown stacked upon each other. Okay. So this is also called as Golgi stack. Okay. And these are Golgi cisterna. Now this Golgi complex can associate with many other numerous Golgi complexes and form what is called as Golgi ribbon. Okay. So here in Golgi ribbon, these are the connecting structures which are called as tubules. Now this Golgi ribbon is formed when the cell is not dividing. But this Golgi ribbon is going to undergo a deassembly when the cell prepares to divide. And in which phase of cell cycle does the deassembly of Golgi ribbon takes place? G2. We know that cell cycle progresses from G1 to S to G2. So in G2, the Golgi ribbon dissociates or undergoes a deassembly. Why? Because of course, every other cell organelle except mitochondria and lysosome, like endoplasmic reticulum, like microbodies, the cell doesn't require when it is undergoing cell division. So there is a disorganization or deassembly. In the same way, the Golgi ribbon also undergoes deassembly. And there is a checkpoint to confirm this, which is called as G2 Golgi ribbon checkpoint. So at the G2 phase, the Golgi ribbon should disintegrate itself, and this should also be confirmed. And cell confirms it through G2 checkpoint. So then why did this organize? This organized because the secretory proteins which are required for formation of spindle fiber and the regulation of those proteins during cell division is of paramount importance. Because the protein which is required for organization of microtubules would be tubulin, and this is ultimately processed from Golgi ribbon or Golgi complex. Then once it is formed, then cell can afford to disintegrate the Golgi ribbon. Why is Golgi ribbon organized? Just because the requirement of the protein may be very high. So, of course, a single Golgi complex would not suffice for the cause. That is why we have Golgi ribbon. So, how interesting, cell aggregates its different cell organelles and makes up a complex altogether to carry out its functions, and then it is disorganized when it is not required. So, this is all about Golgi ribbon. And if you are interested in such content, please subscribe to the channel and read the channel description. In the channel description, I have put up a link on my book Curious Corners in Biology, where you will find all such inquisitive, scientific, juicy questions in biology, which a reader, while reading textual content of biology, naturally comes up with certain questions in his or her mind. So I'm trying to address such questions. If you have yours, you can give your inputs as well. So that much for today. Thank you very much. We will be soon seeing you with bunch of other interesting questions. Till then, bye.